Hey, welcome to our Zoom meeting. My name is Aubrey Miller. I'm an incoming freshman in the class of 2027, and I will be majoring in biochemistry.
Hello, my name is Hannah Rowe. I'm an incoming freshman in the class of 2027, and I will be majoring in genetics.
Our research project mentor was Dr. Cheryl Ingram Smith. She works in the genetics and biochemistry department. The name of our project was called The Metabolism and Cyst Formation in the Human Pathogen Entamoeba histolytica.
Our project was gene analysis of the metabolism and cyst formation in the human pathogen Entamoeba histolytica.
Important terms to know includes amoebic dysentery, which is a severe form of amoebiasis associated with stomach pain, bloody stool, and fever. Incystation, which is the formation of three-layered hard crust or cysts around the amoeba to protect itself from unfavorable conditions. E. histolytica is a single-cell parasite that is transmitted to humans via contaminated water or food.
Entamoeba histolytica is a parasite that causes dysentery. According to the World Health Organization, there are about 100 million symptomatic E. histolytica infections each year. Although up to 1 billion people may be infected, as 90% of infections are asymptomatic.
Amoeba histolytica enters the body predominantly through your mouth, which then goes through your GI system, causing the infection. This is done through contaminated food or water. The cysts, which causes infections, are spread through feces.
E. histolytica has two life forms, the growing amoeba and the dormant cyst. Cysts convert to the amoeba form in the small intestine, which then colonize in the large intestine. Here, amoeba can form cysts again in a process called incystation. Incystation requires changes in gene expression. Our research is about genes that are turned up or down at the same time, being regulated by the same proteins binding upstream at specific sequences. The purpose of our study was to look at these genes turned up or down and see if there are any similarities or points of comparison.
The method we used for this research was through a program called AmoebaDB. We were given a set of genes, which were then entered into the AmoebaDB program, which analyzed the genetic sequences used to put inside the ME program to analyze the upstream region and sets of genes that were turned up or down together to identify sequences that were shared among multiple genes within the set. This may be a way to identify regulatory sequences responses for initiating the incystation process.
The following were some of our results. Gene EHI 056350, EHI 189190, and EHI 180130, while all are not identical, are all a part of the same gene family. Further, these genes are all hypothetical proteins. EHI 134140 and EHI 120570 are nearly identical and are part of the same gene family, BSPA. EHI 184260 also contains similarities to the gene EHI 120570 and was found to be in the same BSPA gene family. All three of these are leucine-rich repeat proteins and are protein-coding genes.
EHI 157010, also known as alcohol deoxygenates, and EHI 039190, known as aldose reductase, were almost identical. They were from the same gene family, which explains the results. EHI 041608, known as mucin-like protein number 1, and EHI 067700, known as protogen 04, has different p-values. However, their motifs show homologous sequences. With further investigation, it was later discovered that they were in fact from the same gene family as well.
So from this research, we can continue to examine aldose reductase and other gene sets with multiple identical genes to determine whether they have similar promoter structure or are differently regulated. E. histolytica BSPA gene family is a large family, and only 3 of these genes are unregulated. The next step would be using the sequence motifs to search the genome to find what other genes belong within this family. This might give a clue as to which ones are most important for regulation. Ones that are present for tons of genes might be general promoter sequences, but ones that are less frequent might be binding sites for proteins that cause up or down regulation in response to certain signals, such as incestation.
Finally, we would like to thank the Clemson's Honor College and the EUREKA 2023 online undergraduate research program for giving us this opportunity. In addition, we would like to thank our mentor, Dr. Cheryl Ingalls.
Thank you.